Now, good morning, students. Today we are going to study about chromosomes, and these chromosomes are first studied by Walter Fleming, a German scientist, in 1882. So, the topics that we are going to deal with are definition of chromosomes, its location, types, function, structure, and of course its ultrastructure and DNA replication. In definition, the chromosomes are highly filamentous colored bodies that are found in the nucleus, which are visible during cell division and carry hereditary units or genes. Now, what are these genes? They are nothing but specific sequence of nucleotides of DNA strand on a chromosome responsible for a hereditary characteristics. The chromosomes are located in the nucleus of the living cells. The function of the chromosome is to carry hereditary units, and the function of the genes is to transmit the heritable features from parents to their offspring. Now we come to the types of chromosomes on the basis of the location of the centromere and on the basis of the function of the chromosomes. Now, on the basis of the location of the centromere, this is the centromere, this is a single strand of chromosome, and this is a duplicated strand having two sister chromatids. Now, the centromere here joins the two sister chromatids, and the centromere also joins the chromosome to the spindle fibers during cell division. So, sister chromatids are only found or observed during cell division stage. Now, each chromosome has two arms, P arm and Q arm. Now, on the basis of the position of the centromere, there are four categories of chromosomes. The one where the centromere is present exactly at the center, dividing the P arm and the Q arm into equal lengths is known as metacentric, whether it appears or acquires the shape of V during cell division or anaphase stage. And the next is submetacentric, in which the centromere is slightly above the center, so the P arm and Q arm are of unequal lengths. And this is in this stage, the submetacentric. In the anaphase stage, it takes up the shape of L. Acrocentric chromosome is where the centromere is slightly below the proximal end. So it has the P arm is very small than the Q arm. And this acrocentric chromosome acquires the shape of J during cell division in the anaphase stage. The last category is the telocentric, where the centromere is present at the proximal end, and this chromosome takes up the shape of I in the anaphase stage. Now we come to the basis of the function of the chromosomes. On that basis, the chromosomes are categorized into two categories, autosomes and allosomes. Autosomes and allosomes we find in human homologous sets of chromosomes. You can see the entire set of human chromosomes are 23 pairs in a diploid cell, and this displays the karyotyping of the chromosomes. And these first pair, one pair has two chromosomes, which is a homologous pair. Each one is obtained from each of the parent. Now 1 to 22 chromosomes, pair of chromosomes, they carry heritable features that are responsible for body features, and they are called as autosomes. The last pair, the 23rd pair, is allosome or sex chromosome, which is responsible for determination of the sex of the offspring. And here, when the two chromosomes are similar in size, that is X and X, the offspring is determined to be a female child. But when there are dissimilar pair, X and Y chromosome, which is much smaller, then it denotes a male child. So on this, there are these particular different types of chromosomes. Now we come to this condensed structure of the chromosome, which is visible only during cell division. And as you can see, each chromatid here, which is condensed to form a pack of chromatin fibers, they are enclosed by a protein core. Now these chromatin fibers, when they are open, they look like a pack of noodles, but when it is opened, you can see this coiled and supercoiled structures having these units or the subunits, which are called as nucleosomes. Each nucleosome, this is one nucleosome, so there are billions or trillions of nucleosomes that form the chromatin fibers. Now they are condensed in the sister chromatid or in the chromatids. Now these histone proteins are 8 in number. The histone proteins are those protein molecules that are specifically found in DNA because they help in packaging and calling of the DNA strand into the nucleosome. And these strands are called the DNA. Now, if this DNA is opened up, you find this macromolecule or double helical structure of DNA. This particular structure of DNA actually was founded or worked finally by Watson and Crick in the year 1953. Now we come to the Now we see here the double helical structure of the DNA. DNA is a deoxyribonucleic acid which has two antiparallel strands. Now there are two nucleic acids, DNA and RNA, in the nucleus. Now the RNA has a single strand and DNA has two strands that run antiparallel to each other and form a ladder-like structure. And the difference between them is RNA is single strand and DNA has double strands. And the DNA has in the rungs of the ladder as the nitrogenous bases which are arranged in this manner complementary to each other with thymine and adenine and guanine and cytosine. But this arrangement is complementary to each other because there are purines and pyrimidines. But in RNA, in place of thymine, the nitrogenous base is uracil. The rest of the nitrogenous bases are same. Now we come to the ultrastructure of the DNA. Here how these nitrogenous bases are arranged, how they form a complementary base pair. Now look here. This is a double helix of DNA which has these two strands like a ladder that is wounded to form this coil structure. But if you open this coil, we find this DNA ladder. And in this DNA ladder here, we find that the sugar molecules and the phosphate molecules forming the backbone or the banister of the ladder. The rungs of the ladder are made up of the nitrogenous bases that I told you about. But they are complementary. C and G are complementary of cytosine and guanine. So cytosine is purine and guanine is a pyrimidine. Now you can see here also purine and pyrimidines. They are joined by 3 hydrogen bonds. So the bonding between them is with the hydrogen bonds that joins them together and prevents them from getting separated. And adenine is paired with thymine with 2 hydrogen bonds. So thymine and adenine, purine and pyrimidine form another complementary base pair. And each unit of this DNA molecule is a nucleotide that we saw in the gene definition. The nucleotide has 3 components. One 5-carbon compound, pentose sugar, and a phosphate group. This phosphate joins 2 pentose sugars and keeps them in position. And these 2 banisters are anti-parallel to each other. They are joined by these nitrogenous bases on the inner side, forming a complementary base pair. So this complementary base pair, when the hydrogen bond dissolves between them, they get segregated. And when they get segregated, these units or the nitrogenous bases that can be found in the nucleoplasm can be collected and a new strand can be formed on the parental template. So these 2 strands will serve as the parental template on which a new strand of DNA can be formed, right? So let's see the DNA replication in the. Now we are going to observe DNA replication, right? In this DNA replication, you see this enzyme helicase is separating the 2 strands by dissolving the hydrogen bonds between them, between the nitrogenous bases, and keeps them apart. Now these 2 strands are the templates, parental templates, on which the DNA polymerase in this leading strand is adding the nucleotides in the direction of 5' to 3' direction. But the other blue template or the blue parental strand, here you see this is the lagging strand, and the polymerase doesn't directly attaches the nucleotides, but here the RNA polymerase is an enzyme which leaves a RNA primer, so which initiates. Now see this, the RNA primer and RNA primase leaves the RNA primer here on the lagging strand, and the DNA polymerase 3 attaches the nucleotides in the direction of 5' to 3' direction. Then DNA polymerase 1 replaces the polymerase 3 and leaves another segment of nucleotides in the direction of 5' 3' direction. Now this is the DNA ligase which joins the gap and seals it. Now this helps in coiling and the joining of the nucleotides to the lagging strand. Now you can see how discontinuously in the lagging strand the nucleotides are attached. But in the leading strand, as you can see, the DNA or nucleotides are attached in the leading strand continuously. Now this is how DNA replication occurs, which leads to the formation of two new strands from one single parental strand. So when the DNA forming two new strands or DNA replication is occurring, which are exactly similar to the parent strand, in the similar manner, the chromosomes also get duplicated because the ultimate structure of the chromosome is the DNA. Now this leads to the formation of two sister chromatids, and that help in the finally in the formation of sister chromatids that leads to the chromosomal equal number of chromosomal distribution in the daughter cells during cell division. I hope you have understood, children. Thank you. Have a nice day.